I'm Daniel Levine, and this is the Bio Report. Senescent cells, ones that no longer divide but are metabolically active, are associated with aging. They're also implicated in a broad range of aging-related diseases, including cancer, diabetes, and neurodegenerative conditions. In the case of cancer, these cells can help protect tumors from a person's immune system. Immorta Bio is seeking to address aging-related diseases by targeting senescent cells and killing them. We spoke to Thomas Ichim, President and Chief Scientific Officer of Immorta Bio, about aging-related diseases, the role senescent cells play in these conditions, and why the company's therapeutic approach may also have promise of addressing aging itself and extending healthy years of life. Thomas, thanks for joining us.
It's my pleasure to be with you.
We're going to talk about aging, Immorta Bio, and its platform technologies. Let's start with aging itself, which Immorta says it treats as a disease. What's the case for thinking of aging as a disease?
Well, I think probably the biggest thing is that aging is infectious. So, about 10 years ago, 15 years ago, there was a revolution in the way we see aging. Historically, aging was seen as accumulation of different mutations, accumulation of damage. Then once the cells reach a critical threshold, it basically becomes senescent. And the idea was that senescent cells just hang around and do nothing. Then about 15 years ago, Dr. James Kirkland started this idea that the senescent cells are actually pathological, that the aging cells are not only are they old cells, but they accelerate the aging of the cells around them. So specifically, this led to a revolution, which was shown that if you kill the cells that are senescent, the cells that are left over actually become younger. So it seems like the aged cells have an infectious nature where they tell the cells around them to grow old. And proof of this infectious nature of aging is that when you take cells that are senescent and you implant them into mice that are middle-aged, those middle-aged mice get accelerated aging. On the flip side of the coin, if you use what's called senolytics, which are agents that kill senescent cells, then as I mentioned, you get regeneration. So the idea of aging being a disease is that aging is actually appears to be at a cellular level an infectious process. And if we can reduce the number of these aged cells, then we have a shot at attaining a more profound regenerative activity in a human or in an animal.
And for listeners not familiar with the term senescent cells, can you describe what that means?
Well, so these are cells that this whole idea originally came back from a guy named Hayflick more than 100 years ago, who came up, who saw that when cells multiply more than 50 times, they stop multiplying, and it all depends on the cell type. But in general, the majority of the cells, they continue living, but they're not dead. They're metabolically active. Basically, a senescent cell is a cell that is multiplied more than 50 times, which is called replicative senescence. Or you have stress-induced senescence, which you can see, best example of that is photoaging. People who, especially here in California, who've been out in the sun too much, and this kid gets old just because of the stress. So a senescent cell is a cell that's alive, not dead, and is basically an aged, dysfunctional cell.
My sense, though, is that we've discovered these senescent cells have some biological function, that in some ways they're protective against cancer. There have been efforts that to remove these cells and replace them with transfused blood, and that doing so actually had some negative consequence.
Well, so this is where we have to be very careful. So the process of senescence the process of senescence protects against cancer in the sense that, let's take for example a skin cell, a melanocyte. So a melanocyte is exposed to UV radiation. That causes DNA damage, right? The DNA damage tells the cell, hey, become senescent. Senescence is a protection against cancer because instead of multiplying indefinitely, the cell reaches cell cycle arrest. Does that make sense? So you have, for example, tumor suppressor genes like P53. So P53, when your melanocyte is exposed to the sun and there's DNA damage, there's DNA damage sensing enzymes that activate P53. P53 tells the cell, hey, you got to stop multiplying, you got to become senescent. And that stops the cancer. P53 induces senescence. And a fact that you might be interested in is the reason why elephants don't get cancer. Cancer is extremely rare in elephants, is because elephants have like 20 copies of P53. So the senescence process protects against cancer if you're talking about at the level of the cell itself, of the mutated cell. But quite the opposite, the senescent cell surrounding the cancer makes cancer grow faster. And what I mean by that, the evidence of that is some of the data that we'll talk about in this discussion, but other people have also published that in various cancers, if you give a senolytic, you actually sensitize the tumor to radiation, chemotherapy, or immunotherapy. And that's because the senescent cells that surround, so the cancer is producing inflammation. The inflammation causes accelerated aging surrounding the tumor. That accelerated aging causes senescent cells, and those senescent cells produce factors that in turn protect the tumor or suppress the immune response to the tumor. Now, in terms of transfusing young blood, here's the thing. Anything that causes regeneration can cause neoplasia or cancer. So, we hear this all the time, people saying, oh, let's take stem cells because we have cancer. And, I've been told not to use colorful language, but I really have to use colorful language. That's probably one of the most idiotic things I've ever heard, is using stem cells to treat cancer, to treat solid tumors, because stem cells by their very nature, and when I say stem cells, I mean hematopoietic or mesenchymal, both of them stimulate production of new blood vessels, a process called angiogenesis. In the field of oncology, in which we've published a lot, you want to stop production of new blood vessels. So, you do not want in an oncology patient, you do not want regenerative factors, because regenerative factors usually stimulate carcinogenesis. Does that make sense? Did I explain it correctly?
No, I follow it.
Okay.
How does this then inform your approach to drug discovery?
So to drug discovery, basically, well, let me put it this way: we primarily see the cells as a drug. We are a cell therapy company. Everything we've done has been in the area of cells historically. But Dr. Resnick and myself, we have made companies in the areas of stimulating regeneration. We had a company called Medistem that then we sold to a New York Stock Exchange company called Intrexon, where we found a new type of stem cell. We have a company, Batu Biologics, where we use a cellular vaccine to kill new blood vessels. Our drug discovery, how the process of senescence guides our drug discovery, is basically our drugs are cellular therapies. And our drug discovery, first of all, and we're going to talk about this, is we have, I guess, two major platforms. The first platform is cellular rejuvenation, making new cells, regeneration. The second one is called senolytic immunotherapy, using the immune system to kill senescent cells. Basically, we are cognizant of the process of senescence, and we are also cognizant of the process that senescent cells actively suppress regeneration. So we have used dendritic cell technology. So dendritic cells are the most powerful immune stimulator the most powerful immune stimulatory cell that exists, or dendritic cells. And we have used them to successfully induce immunity against senescent cells in a patient-specific manner, against senescent cells that are surrounding and protecting the tumor. That approach could be useful and could be applied in the future to increasing therapy of our cell-based stem cell approaches.
Let's talk about the platform technology. Walk us through the process of how you obtain these cells and what you do to turn them into what you call your revivify therapies.
So basically, what we do is we take a blood sample, and from the blood sample, basically about a tablespoon of blood, and we have identified cells within the blood that are more amenable to dedifferentiation. So basically, what we do is we take blood cells, and we call these cells amenable stem cells, ASCs. We isolate these ASCs that are amenable to being made into early stem cells. Then what we do is with our proprietary cocktail, we make them into what we call PRC, personalized regenerative cell. The PRC is basically like an iPSC. So iPSCs these are immortal cells, cells that can multiply forever. They can fix themselves. They have telomerase. They have enzymes, high levels of enzymes that fix and degrade proteins that are non-functional. So basically, we generate a patient-specific cell. So everything we do is autologous. A patient-specific cell that is immortal, and that's basically patient-specific iPSCs. The reason why we call them PRCs is because we generate them using a proprietary methodology, which allows for a more rapid generation of this dedifferentiated cell and more economical. See, the thing is, the anti-aging, everybody talks about anti-aging. Well, Yamamaka discovered that you can take an adult skin cell and make it all the way dedifferentiated with 4 proteins back into a cell that can make any cell of the body. The problem is that technology, while it's done amazing things for drug discovery, the clinical translation has been somewhat hampered, primarily because of cost of personalized cell manufacturing. So one of the first things that our company has done is to reduce the cost of this pluripotent cell generation in order to be able to allow it to be personalized. Well, that's the first platform. It's called PRC. The second platform is called the personalized progenitor cells, PPC. And these are cells that can differentiate into different cell types of the body that are therapeutic. So we have, for example, a PPC for hepatocytes, and we have performed preclinical studies. We're in the process of getting ready to file an IND for that. So PPC technology is to differentiate the PRC into specific tissue, and we've been able to do this for a variety of tissues. Liver is our main one. We have generated insulin-producing cells. We have generated neurons, specific types of neurons. So the other platform is the PMSC platform. So instead of taking the pluripotent iPSC type-like cell and making it into specific tissues, in this case, we make it into mesenchymal cells. That one we call the PMSC. And the PMSC is very excited because we can make younger mesenchymal stem cells that are personalized. You see, the big problem with mesenchymal stem cells in the field has always been that the mesenchymal stem cell, if you use it from the same patient, it doesn't have as much activity because in the autologous situation, the patient you're treating usually has comorbidities when they're adults, which reduces the activity of the cell. If you use allogenic cells, the problem with allogenic mesenchymal cells, even though you can obtain younger ones, those ones end up getting rejected because they're allogenic. They're eventually rejected. So with our PMSC technology, we can make autologous mesenchymal stem cells, but we can make them at a younger age so that they have more regenerative factors.
You're delivering not just the cells, but you say a cocktail. How are they delivered to a patient, and how do they get to where they need to go within the body to be effective?
So the cells contain they are optimized for what's called CXCR4. This is a homing receptor. So any kind of damage in the body, the majority of damages in the body are associated with hypoxia. So somebody you fall down and you hit yourself. First thing that happens is the blood coagulates. The blood coagulation activates hypoxic-induced genes. One of the hypoxic-induced genes is called SDF1 or CXCL12 is the name. And then this binds to a receptor on the mesenchymal cells called CXCR4, and that's what causes the homing. So we know this from a variety of standard mesenchymal stem cells, and there was work done out of the Mayo Clinic, really good work where what they did is they would put into a damaged tissue, they would just put the homing signal, the SDF1. They had SDF1 gene therapy. They would inject it in the heart and different organs, and that would just call stem cells. So it's a well-known phenomenon that stem cells are home to CXCR4, and home to SDF1 via CXCR4 on the stem cell. And what we do is we optimize the PMSCs for these homing signals so that in a lot of the situations, you just inject them intravenously and they find their way. That's another thing that, a lot of people haven't realized is the stem cells are not a magic bullet. Everybody talks about the stem cells. How many drugs do you know that you take only one pill? Pretty much none. There's very few and far between. And the problem is a lot of the stem cell clinical trials do one injection, one infusion or two infusions. This is why a lot of times, we don't see a lot of the stem cells historically have done excellent phase 1 and 2. And then when they get to phase 3, they end up flopping. And one of the reasons we believe is that not enough attention is paid to the dosing regimen. When you give stem cells IV, a lot of them go to the lung. If you give more than 100 million cells, a lot of them just go to the lung. So, one of the things we've been very involved in and because we have experience both from owning stem cell companies and also from owning the company BioRazi, which has done multiple stem cell clinical trials, we understand there's a lot of subtleties. You can't be a bull in a china shop. And so one of the things is dosing is very important, the route of administration, the patient characteristic. If you throw seeds on gravel, the seeds are not going to grow well. There's no reason why stem cells should be any different. We include in our IP and in our work that we're going to take to the clinic, we include different recipient preparation techniques to make sure not just that the stem cells home, but once they get there, there is a place waiting for them, a niche waiting where they can grow and prosper.
Let's talk about the pipeline. You're in preclinical development on all your therapies. Revivify program in liver failure is your most advanced program. What's known about these therapies from preclinical studies done to date?
The most advanced program is our cancer one, because the oncology one, we have an IND filed and an IND number has been granted. But the liver is very advanced. And look, what we know is we have animal data for the liver. We have animal data in 2 of the major animal models, the carbon tetrachloride model and the acetaminophen model. We have a mechanism of action for which we filed intellectual property. Mechanism of action is very interesting. It's called interleukin 35. So it's funny for the immunologists because everybody knows up to interleukin 12 or 15. Now we're all the way up to 35. But interleukin 35 is a major stimulator of T regulatory cells. And so what we've identified and filed IP on is that in our treatment of liver failure, the PMSCs that we generate and the hepatic progenitor cells, there's a synergy that induces generation of interleukin 35, which causes T regulatory cells. T regulatory cells then go and cause more mesenchymal cells being made, endogenous mesenchymal cells, and this seems to trigger a self-feeding regenerative loop that it triggers it. This is what we see to be inducing the healing in liver. Other people have used stem cells for liver failure before, and we've looked at those studies and spoken to the investigators in great detail, because that's the exciting thing about living in the times we live in now. There is a lot that has been done, and now what we believe is just a thing of optimizing it and putting all the dots together. We're not inventing something brand new. We're just taking what's there to get what's out there and optimizing it.
What's the development path forward for that?
So we plan to file an IND probably for liver probably before the end of the year. For our lung cancer program, we hope to have the IND cleared early next year.
You mentioned your cancer program is the most advanced program. Is this the Sanovax program?
Yes.
And that's a precision targeted immune cell therapy. Walk us through the process of creating a Sanovax therapy.
So with that one, what we do is we take fibroblasts. It's not even fibroblasts. We take a specific type of cell from the skin of the patient, and we expand those cells. We take a tissue biopsy, we isolate a specific cell type, which we found is a lot more sensitive to senescence. We induce accelerated senescence in vitro from the patient's cell type, from the patient's cells. We double check with our quality control to make sure that we've generated senescent cells. And then we take antigens from the senescent cells and we pulse dendritic cells that have been made from the same patient. So basically, your viewers, your listeners may be familiar with the Dendrion. Dendrion was the first FDA-approved immunotherapy. It was an autologous dendritic cell therapy where they put prostatic acid phosphatase, that's an antigen, PAP. They put a fusion protein of PAP with GM-CSF to make dendritic cells, and these dendritic cells stimulate immunity to prostate. The company Northwest Biotherapeutics takes glioma antigens. Basically, they do a surgical biopsy of the glioma. They generate dendritic cells and pulse them with glioma antigens, and they've had very good responses in people who had months to live, who are living 5 years. That's called Northwest Biotherapeutics. We said, hey, if people can do this with oncological targets, why can we not induce a similar polyvalent vaccine targeting senescent cells? And we previously, Dr. Resnick and myself, we previously have an IND cleared in our other company, Batu Biologics. We had an IND cleared using the same approach to kill tumor blood vessels. So we modified that approach to kill senescent cells. We demonstrated in vitro and in vivo that we can kill senescent cells, and that killing of senescent cells results in regression of non-small cell lung cancer. So we took that data, did a lot of tox studies, went to the FDA, received an IND number, and now we're in the process of negotiating with the FDA and moving forward to the clinical trial.
It's interesting that you're developing a cellular immunotherapy, but you're not targeting the tumor cells. You're targeting the senescent cells. Does that end up enlisting the immune system to attack the tumor, or does it just prevent the senescent cells from masking it in some way?
What it does is removing the senescent cells exposes the tumor to the underlying immunity. And so for this first phase, we're using them as a monotherapy. And we've seen responses with the monotherapy, with the Sinovac alone. We've seen responses. We have seen responses to the monotherapy in the animal models. We believe we're going to see responses in the humans. Once we have a baseline of how the monotherapy works in the humans, then we do plan to move on into combinations with things such as checkpoint inhibitors and standard chemotherapy.
It would seem that, given this unique mechanism of action, it would make sense to do that. Is that
exactly? But we want to walk. We want to walk before we run. We want to first of all define the parameters. Nobody has created a clinical product where you're killing senescent cells. And while this is exciting because typically you think of side effects of cancer drugs as being negative. The side effects here might be regeneration in other parts of the body because you're inducing a systemic immune response to senescent cells. But we need to be very careful because this is a game-changing therapy, and it could alter the face of oncology if done appropriately. But if not done appropriately, we all remember what happened with gene therapy when the first gene therapy trial did not end so well. So every time you do something game-changing, and the thing is, everybody in this company has been around for a long time. We've done a lot of clinical trials, and we know a lot of times when you do something, it's a lot better to be conservative and cautious and have a lot of respect for the new science as opposed to being a cowboy. And then, you might have short-term consequences, but long-term you don't win.
Is the expectation that you'll not only be able to treat diseases of aging, but be able to treat aging itself and expand healthy lifespans?
Yes, that is the overall goal of the company. But obviously, it's very difficult. As you can imagine, it's very difficult to do a 30-year or a 20-year clinical trial, right? And we don't think our investors want to wait that long. So the initial idea is to dip our feet wet, understand the process, look at situations where the aging is accelerated, such as in neoplasia. Look at ways of inducing regeneration, and then eventually go for the overall aging. You have to realize that. I mean, obviously, you know this, that with any drug development, there's a risk and a reward, and to be treating all people who are older with a therapeutic intervention such as Senovax, you want to make sure you have a lot of safety data. Same with our regenerative medicine platform. So you heard about people injecting other people with young blood. Well, we can basically make a younger version of yourself. It's been published that young MSCs, so in animal models, the beautiful thing about animal models is that the strains of mice are all inbred. And because they're inbred, they're genetically identical. So there are studies published where you take mesenchymal stem cells from a young mouse, put them in an old mouse, and the old mouse starts becoming younger again. We could ideally do this in people with our technology. There's companies like Longeveron who have done mesenchymal stem cells and have shown some level of efficacy signals, not as much as we all wanted to see, but they have shown some levels of anti-aging effects from mesenchymal cells. Our ability to make autologous younger cells, we believe, are going to give us a lot more advantage to what's been done before. But that being said, our main initial excitement is towards this terrible disease called cancer, and also towards this terrible condition called liver failure. As you know, in liver failure, there's a large number of people that die on the transplant waiting list. The thing about liver failure that makes us very interested in is that it's already published that if you inject hepatocytes by themselves intraportally, you can save somebody. It's just you don't have enough hepatocyte cells because you don't have enough donors. Well, we've generated hepatocyte cells in large quantities, and that's what makes us very excited, is because it's already published that the hepatocytes can help this condition. We just have an alternative source of hepatocytes.
If you do eventually pursue aging as a disease for a therapeutic, what do you think about in terms of endpoints? What might an endpoint that the FDA would accept look like for a study in that?
Muscle frailness, chronic inflammatory cytokines. So, for example, high sensitivity, CRP, different mental function tests, things like that.
And how about payers? How do you expect payers to view such therapies, or would patients need to be self-funded?
Well, we're going to for the cancer and for the liver failure, we are following just a very standard drug development.
I'm thinking in terms of aging as the disease you're treating. If that were to come, how might payers look at that?
Well, with that one, it depends how potent the efficacy signal is. Because if we have a potent efficacy signal, I believe that there will be a support. If it's something that's very patient specific and that's a little bit up in the air, as you can imagine, we probably will not get as much support.
And how is the company funded to date, and how far will existing cash take you?
Well, so right now the company has done a pre-seed round and is funded by the founders. It's also funded by it's also funded by the fact that we all put a lot of work into it and none of us are taking a salary. And so we are in the process of doing a seed round. So first of all, the cash we have now is going to take us forever because we all work for free. And we all put the money into it. All of us have had successful exits in our business. So nobody is starving, if you know what I mean.
Well, but that's not going to fund a clinical trial.
Well, no. But actually, it might. We have self-funded trials before, but right now we are doing a seed round, and that seed round, the use of proceeds is to fund the clinical trial. So from that point of view, we are very fortunate to have the resources we have and the colleagues and collaborators we have, and I guess in a way it's a very unique company because it's a mission-oriented company. All of us have this burning desire to solve this problem, and all of us have had successes in the biotech and pharmaceutical industry. For example, our chief medical officer was the head of medical affairs at Pfizer, and so on. So we all see this as a project of passion, and we are very secure in the financial future of the company, regardless of which way the markets turn. As you've seen, there was somewhat of a bloodbath in biotechnology, in the stock market at least, but we're very fortunate to have some protection from that.
Thomas Ichim, president of Immorta Bio. Thomas, thanks so much for your time today.
Thank you so much. This was very enjoyable.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.